Good morning, good afternoon, everyone. We'll give you all a moment just to get settled, and we will begin in just a moment. Hello, everyone, and welcome to today's webinar titled Optimizing RNA Manufacturing for Rapid RNA Therapeutics Development. Today's webinar is a part of the 11th annual event in the Cancer Research and Immuno-Oncology Week virtual event series. I'm Christy Jewell of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and sponsored by GenScript. For more information about our sponsor, please visit genscript.com. Now, before we begin, I do want to remind everyone that today's event is interactive, and we encourage you to first participate by communicating with other attendees using our live chat feature during the presentation. You can find that live chat located on the left of your screen. You may also participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We will answer as many of your questions as we have time for at the end of the presentation. You will also notice that the speaker has provided a survey located on the right of your screen. Please take a few minutes to answer the survey questions. It will be available both during the live and the on-demand period. Now, if you have any trouble seeing or hearing the presentation, click on the Help Desk button located at the top of your screen within the event. Now let's get started. I would like to now present today's speaker, Fengmei Pi, Head of RNA Biology, GenScript. For a complete biography on our speaker, please find it on the left of your screen. Fengmei, welcome. You may now begin your presentation.
Thank you very much for your introduction. I'm going to talk today about how to optimizing RNA manufacturing for rapid RNA therapeutics development. My talk today will cover about the optimizing of making Cap1 RNA, Cap2 mRNA, and the circular RNA. So GenScript has a long history in making customized gene synthesis and plasmid preparation. So we launched the customized mRNA synthesis and circular last year. And this year we're introducing circular RNA synthesis and LNP formulations for assisting customers' fast delivery of development of RNA-based therapeutics. mRNA has different kind of capping options. There's a lot of research regarding this. Cap0 mRNA is usually found more in the plant-based RNA or the virus RNA structures. Cap1 is the main mRNA structure that's found in mammalian cells and is also the currently used vaccine version for COVID. That has been proven well for the human therapeutics development. And recently, I also want to introduce about Cap2 mRNA. So Cap2 is also a natural form of RNA that is found in nature, and its function is under discovery. But there is a report showing that Cap2 RNA has a better response at immune stress condition, and it also has more dominance at the elder cells. It also impacts the protein production in a cell-specific manner and contributes to RNA immune evasion. So this will be the structure of how the Cap1 mRNA and Cap2 mRNA look like. In general, the Cap1 mRNA have the first base after phosphate backbone being methanated, and Cap2 mRNA usually have the first two bases methanated at the ribose. The mRNA customized mRNA service we're introducing at the GenScript will include the following composition and properties. The mRNA will include the 5' cap structure and the 5' UTR and your ORF sequences and 3' UTR and poly A tail, as you are already very familiar with. Here we can offer the customized mRNA synthesis from scale of 0.2 milligram to 200 milligram or even more that we're going to launch next year. And about the cap options, like introduced, that we can offer all kinds of caps, including cap 0, cap 1, and newly we're announcing the cap 2 mRNA service. So poly A tail wise, we have vectors that has poly A tail built in with either 100A or 120A. And yeah, you can also customize this with different poly A numbers. About the modification, that's quite important. It has been found that the unmodified U would trigger a lot of the immune response in the mRNA therapeutics. As for the N1-methyl-pseudo-U or 5-methoxy-U are commonly used modifications that can help to reduce the immune response and also enhance the mRNA expression by improving its stability. Another combination is by pseudouridine and 5-methyl-C is also being validated, very helpful in improving RNA therapeutic effect and reducing immune response. So about the purification method, when the RNA preparation scale is less than 10 mg, we just use the chromatography method to purify it. And when it is more than 10 mg, we prefer to use lithium chloride precipitation, since this gives the highest quality of the mRNA at larger scale. And we also have the preclinical grade RNA. With that, we do the oligo-DT purification and also optionally double-stranded RNA removal, which will help to give you the highest purity of RNA when it's a long one and get minimum residue of double-stranded RNA, which is also a source of triggering immune response. And we are also offering a full spectrum of QC test that will introduce later. So the process of making mRNA will be like this. We will start from plasmid linearization. So the circular plasmid will be our key starting material. After receiving that, we will linearize it with the choosing restriction enzyme and then purify the linearize the plasmids and then put them into the in vitro transcription reactions. And then that will be purified by the column-based method or oligo-DT method or the double-stranded RNA removal purification, which is also chromatography-based method. And then we'll move on with the final QCs. So start from the beginning. The design of the mRNA is very important. We'd like to introduce that the GenSmart is a very good tool to optimize your ORF sequences. It has a good algorithm for codon optimization that can help to optimize the protein expression with that mRNA. And it has been validated, patented with more than 200 factors screened in this algorithm development. And there's a paper published showing that using GenSmart's algorithm, optimize the sequence, have the best results during the vaccine development and other previous researches. Yeah, next I would like to introduce the key features that's impacting mRNA quality and efficacy. So the first thing, very importantly, would to think about the integrity, which is the mRNA purity. So here we tested the integrity of mRNA by bioanalyzer, which is a type of capillary electrophoresis. Using our optimized in vitro transcription condition and purification process, we could reach a purity of more than 95% of the RNA for the EGFP. And comparing with competitors' products from company A or B, you can see we have a much higher purity. In results, we got a better expression, which is about 4 to 5-fold higher in the EGFP case. And Also, so ESP-Cas9 mRNA is another case we studied, which is 4.5 kb mRNA. Using our process, we could reach a purity of higher than other companies, and in return, it will translate into higher expression efficiency in cells. So those cells assays are done in the A549 cells in vitro, transfected with lipofectomy, and we tested expression after 24 hours. Second optimization factor for the mRNA production would be the purification method. Like I briefly introduced, we use the column purification base method or lithium chloride precipitation for larger scale for the default. Since we have found that those methods will generate the RNA with the minimum amount of residues, which could be the free NTPs, the protein residue. And secondly, we would like to recommend to add in the phosphatase treatment for the RNA purification step. Since during the co-transcription or in vitro transcription process, you only could generate mRNA with a capping efficiency more than 95%, even though there could be about 3 or 4% of the uncapped RNA, which will start with a 5' triphosphate, and those will trigger immune response in the cells. So we have found that after the phosphatase treatment could greatly impact the RNA for possibly improve its expression and also reduce its NF-kappa B secretion in the cells. Next, for elevate your RNA's quality, I would recommend to perform the oligo-DT purification, which is an affinity-based method to help you to get the full-length RNA with poly A tails. With this chromatography method, you could see at the end we could elute the very pure fraction of the mRNA to get rid of the the amount of the RNA without the tail. So this graph shown here is a purposely experiment to mix with the tailed and untailed RNA. We could see very good resolution here. And thirdly, there is an upgrade of purification of double-stranded RNA removal. It is also very helpful. So double-stranded RNA is a side product that's generated during in vitro transcription process, where the T7 polymerase might just use its own RNA product as a template and keep making it itself and generate the double-stranded RNA impurities. Those double-stranded RNA could cause strong immune reactions and side effects in the animal studies or the in vivo application. So we have developed this chromatography-based method to remove double-stranded RNA and verified this after this purification, you could greatly reduce your RNA, double-stranded RNA residues in your final product. And another factor would like to consider is about the poly A tail quality. So both the length and the quality of poly A tail are very important. Here we studied how the poly A tail length impacts the mRNA expression. We purposely prepared the same EGFP mRNA with different tail lengths and found more than 80 As are preferred for improving its expression efficiency. And with this, we build the plasmid that has the poly A tail built in, since we have found this will generate the highest quality of poly A tails. And at the end, the mRNA poly A tail quality could be detected. We have a method developed here that you do an enzyme treatment and then measure the poly A tail length by either bioanalyzer or LCMS. LCMS will give you more precision results. And with bioanalyzer using the small RNA analysis kit, you have an error range about 20 or 30%. That's due to the detection method. But LCMS will give you quite accurate result of poly A tail numbers. So here we want to show the comparison of how to add poly-A tail. So instead of building the poly-A tail into plasmid, another option is to add poly-A tail by using poly-A polymerase. Here we compared the mRNA prepared using poly-A tails built in plasmid or added by the poly-A polymerase. So here you can see the mRNA from company B in the red trace. This is a tail that's added by poly-A polymerase, and it will show as a flat distribution and very broad. This is something like we don't want to see, and in return you will see very poor result here. But with the GenScript and poly-A, we see a very sharp peak. That means this poly-A in the mRNA are very centered, so they are very they all have about 120 nucleotide poly-A's at the end. With company A's product, you could see a good poly-A tail, but with the same amount of the RNA, you get a lower peak. So I would expect that partial of the RNA has tailed, and there is also some RNA that's been not tailed. In return, this poly-A tail has great impact on the expression. And lastly, I want to introduce about the QC specifications. That's quite important for the mRNA therapeutics development. At RUO grade, we will do default run agarose gel or tip station analysis to confirm the RNA quality, that is the right length and single bands. And we also measure the concentration by UV and make sure it's no endotoxin, less than 10 U per mg. There's other upgrade QC that we offer. So purity-wise, it's important to learn about the capping efficiency and the site-based purity using capillary electrophoresis. Those are all available here. And impurity-wise, we have developed a method to test the total protein residue, the plasmid DNA residue using qPCR method, or the double-stranded RNA residue using the immune blots method. Yeah, that's all about the Cap1 mRNA. Yeah, I'm very excited to introduce a new category of the Cap2 mRNA. I think that is only launched here, available here. So Cap2 RNA is quite important in nature. It's a form that found in the RNA. There is a paper reporting saying that the second base methylation on the 5' nucleotide of mRNA cap will impact mRNA protein production level in a cell-specific manner and contributes to RNA immune evasion. With that, we have developed a novel Cap2 analog, and we can prepare Cap2 mRNA by co-transcriptional method. With this optimized method, we could generate finally the RNA with a purity more than 90%, and the capping efficiency can reach also more than 90%. And the yield-wise, it's about 3 mg per ml for the IVT reactions. And we also studied how the Cap2 mRNA expressed comparing to Cap1 mRNA. As you have seen before, our Cap1 mRNA made here has a very high level of expression compared to others, and we could reach the same level of expression with the Cap2 mRNA here as well. But its expression in cell line are slightly different. We have observed the Cap2 RNA has a slightly better expression in the A549 dual cells, which has more responsive to the immune factors, and it has a lower expression in the THP1 cells in this experiment. And we further look at that how the Cap2 RNA express at the stressed condition. In the results, we have found like in if there are we challenged the cells, A549 cells with interferon alpha. So when there is no interferon alpha present, the Cap1 and the Cap2 RNA similar. Actually, Cap1 expressed better here. But when there is strong interfering alpha condition, you could see Cap2 RNA expressed better than Cap1 mRNA. So this result is the same as published in that paper in nucleic acid research. The authors also observed that Cap2 RNA has a better response, a better expression under immune stress conditions. So say that is a novel category of RNA structure could consider during therapeutics development. Lastly, we also look at its intrinsic immune activity. So here we tested the Cap2 mRNA's immune effects on the A549 dual cell model. And we found with the Cap2 mRNA with proper dephosphorylation treatment, it could get the similar result as the Cap1 mRNA after dephosphorylation. So there is no innate immune response related to Cap2 mRNA. Yeah, that's all about the Cap2 mRNA technologies. And yeah, thirdly, I also would like to introduce about the circular RNA. So circular RNA is a novel class of functional RNA molecules that have the 5' end and 3' end connected as a circle. It has been reported to be able to be for coding proteins or as a non-coding RNA. The research in the circular RNA field is having increasing interest recently, since there is a proven advantages of circular RNA as therapeutics and vaccines, and it's considered as the version number 2 of mRNA, since it has a better stability for longer expression duration and also higher protein expression efficiency, since it can re-engage ribosome with a rolling circle translation. And lastly, it's reported circular RNA LNPs have better thermal stability than linear mRNA. Circular RNA is also explored recently in the application of the non-coding RNA therapeutics, such as microRNA, the protein sponge, or the siRNA, or the single guide RNAs. The circular RNA service we have developed here were mainly featured at using the self-splicing mechanism to prepare a circular RNA, and we also screened optimized one RES for you to help to express the RNA by the RES dependent translation, instead of the linear mRNA, which will be rely on the Cap1 structure to recruit ribosome. The feature of the circular RNA service here will be about the scale of 0.2 mg to 10 milligram per batch production. And we have the CBB3 RES optimized in our vector for you to use, that you can directly send us your ORF and plug in to make the circular RNA. Modification-wise, we have found that no modification make the best result of circular RNA, since we needed to rely on the natural function of the RES and the exon-intron for the self-circularization reaction. And purification-wise, we have developed our proprietary HPLC purification method to generate the highest purity of the circular RNA, which is quite important for its efficacy. And we perform the QC test to look at its purity, and also can test its ID by the RNase R degradation, and also look at the impurities and the safety aspects. You could also customize the RES design. We'd like to introduce a little bit about our feature of the circular RNA that we have done here. So to moving the circular RNA technology to industry, the first challenge we would need to solve is about improving its purity. That's very important for making the drug. So comparing to, we have invented a new method of making the circular RNA with a higher circularization efficiency, and we have filed patent for that. With the publication method by the agarose gel, you can see this middle band here is the crude circular, and we can get around 52% of crude circular using this publication method. But using our optimized method, we could reach a higher crude circular at 70%. And here you can see the main feature is the precursor strand are greatly reduced. And this is the NIC, the circular RNA. To further improve the circular RNA purity, we have developed this HPLC-based method. Within that, we could get a final circular RNA product with more than 90% purity. I would like to stress again the purity of the circular RNA is quite important. And during our study, when we simply follow the publication method, the expression of circular RNA was not as good as expected. And after we perform this purification, the expression of circular RNA could reach equal or higher than linear mRNA. So here's a comparison of the circular RNA to linear mRNA. Over 6 days time course, we have observed that the circular EGFP have a higher expression than linear mRNA and also longer duration. The cumulative expression of a circular RNA is higher than the linear mRNA. Next, we also want to learn how is the circular RNA stable in cells in reality. Is it as expected? So with that, we perform the qPCR assay. We look at we transfected the cells with equal molars of circular RNA or linear mRNA, and then we did a qPCR assay after different time points to see how many of the RNA are remained there. We can see this circular EGFP have a higher amount of the RNA copy numbers existed from day 1, which is 24 hours after transfection, and also it has a slower degradation. In return, the protein expression, so for linear mRNA, you would express stable expression or slightly degrading of lower level of protein expression from day 1 to day 3. But circular RNA, we observed the increasing amount of the protein expression of GFP over 3 days time course. Another question you might want to ask is about immunogenicity, because for therapeutics development, that's also an important factor to consider. So here we tested the immunogenicity of circular RNA in cell models, look at its impact on the secretion of IL-6, interferon alpha, and NF-kappa B. So the circular RNA has a comparable level of immune stimulation with Cap1 mRNA, which is good since the Cap1 RNA has been proven to be good in the drug development. With that, we are confident that the circular RNA service here will help to speed up your circular RNA drug development. Finally, working with GenScript, we have the integrated production process from gene synthesis to the mRNA production, and this will minimize your need to source materials from different vendors and reduce risk of supply chain issues. And we are also experienced in customized manufacturing solutions. Looking forward to working with you, and welcome to let me know if any questions. Thank you very much.
Thank you, Fengmei, for that informative presentation. It now brings us to our live portion of our webinar. My apologies, our live Q&A portion of our webinar. Now, to our audience, if you have any questions you would like to ask, please do so now. Just type them into the Ask a Question box and click Submit. We will answer as many of your questions as we have time for at the end. We will answer as many of your questions as we have time for. Then we have quite a few questions that have already come in, so let's go ahead and get started with this one. What is the maximum length for circular RNA that you can offer?
Thank you. Christy, so for the circular RNA, our current capability is limited to the 5 kb of the total length that include the RES plus your ORF sequences.
Very good, thank you. Our next question. Do you provide mRNA formulation service?
Yes, we do. We have also launched the generic LNP formulation service for mRNA and or circular RNA in September. Yeah. Let us know if anything we can help with. So those will be helpful for your screening project to make sure if your design of RNA works.
Very good. And it looks like we have time for one more question. For mRNA, what purification method do you use for dsRNA removal?
We actually use the cellulose-based HPLC method, since that will remove the double-strand RNA and also keep the good integrity of the RNA. Yeah.
Very good. Thank you, Fengmei. Do you have any final comments for our audience?
No, thank you very much for attending this session. Yeah.
And thank you, Fengmei, for your time today and for your important research. Before we go, I do want to thank the audience for joining us today and for their interesting questions. I'd also like to thank our sponsor, GenScript, for sponsoring today's webinar. Questions that we did not have time for today and those that are submitted during the on-demand period, they will be addressed by our speaker via the contact information you provided at the time of registration. Today's webcast can be viewed on demand for 2 years. That's through October 5th, 2025. Labrador will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, thank you for joining us. Have a great day.
Thank you.